Today we're going to be talking about genetics. If you're in your notebook and you need a title can be genetics. If you have the note packet, you should already have the learning targets. We're going to go ahead and highlight learning targets underneath number 1. So our goal is to be able to determine the inheritance of a trait, apply genetic terminology and use a Punnett square. And today we're just working on what's called monohybrid, dominant and recessive. Okay, so dominant and recessive problems. And then you should be able to calculate the probability of having a certain trait. So let's go to genetics and let's start with some definitions. The first definition you don't need to write down. I'm not going to test you on a lot of these definitions. You'll notice on the learning target sheet, you're going to be responsible for knowing how to solve these problems. Think about math class. When you solve a word problem, you have to know what some of those words mean. And today we're going to talk about what some of those words mean that you might see in some of these word problems that we're going to be doing in genetics. So genetics is the study of how traits are passed down from parents to kids. A long time ago, people were like, hey, we know something gets passed down from the parent to the kid because kids look like their parents. But we don't know what it is. And actually, before they knew that it was DNA, they actually thought it was protein. They thought parents passed protein down to their kids. And today we know that it's really genes and DNA that get passed down to kids. So genetics is the study of all of that. In the word genetics, we see the word gene. So we need to talk about what a gene is. A gene is a section of DNA that codes for a protein. You'll notice I have a picture of a chromosome here. And we know we have 46 chromosomes in each cell in our body. A gene is a section of that chromosome. Remember, each chromosome's got thousands and thousands of genes. Okay, so if we look at that DNA, this chunk of DNA, this section of A's, T's, C's, and G's, might code for one thing. It's called one gene. And this section codes for another. That's another gene. So we have different genes, okay? Just a section of DNA. We know that genes have a start codon and a stop codon. So within this chromosome, there are lots of start and lots of stop codons. Now, traits, genes code for traits, right? We know that as a result of our DNA, we have traits that you can see, like we have specific eye colors or hair colors, or we're tall or we're short. Even some of our personality things are traits. So a trait is just a characteristic, and oftentimes it's determined by our genes. We're going to also learn though that it's determined not only by your genes, but also by your environment. So ultimately, genes code for proteins, which code for traits. If you're in your notebook, just write this bottom section right there, okay? So we start with a gene. Our DNA has the information on how to make a protein, and that protein gives us a trait. In this example, the protein is insulin, and the trait means we can digest or we can process sugars. If we don't make the right protein, we can't allow sugar into our cells, and as a result, we have type 1 diabetes. If you have brown eyes, the protein is going to be made that actually turns your eyes brown. It's a different protein than if you have green eyes, and that's your trait. So this is just a reminder, nothing to write down necessarily from this slide, but you've got 2 sets of chromosomes in your body. Okay, one from one parent, whether or not you call them mom, or they are the egg donor, or they are your biological parent, or anything of that nature. One of them from one parent, one of them from the other parent, right? So 23 chromosomes from one parent, 23 from the other. So the questions we're really going to be talking about is, well, how do two parents who don't have a genetic disease, why do they have a kid that does have a genetic disease? What happened? We know a little bit about mutations, and that's part of the story, but what if there wasn't a mutation? Or what if a parent has a genetic disease? How likely is it that that kid is going to get the genetic disease? These are the questions that we're going to be answering in this unit. So the first thing we need to talk about are traits. And some of you might remember some of this from 7th grade. We need to talk about genes and traits being dominant and recessive. If you've never heard dominant and recessive, don't worry, it will make sense in a little bit. So dominant is a trait that shows up no matter what it is paired with. We use dominant trait or dominant gene. So you could say a gene that shows up no matter what it's paired with. And we symbolize a dominant gene by a capital letter. Now, when we talk about genetic diseases, some traits, some diseases, if you get one mutated gene, it's dominant, so you have the disease. Some diseases, you need two mutated genes, like cystic fibrosis, because the gene is not dominant. So we use a capital letter. Here's an example here. Dad has the dominant eye color brown. Mom has the recessive blue eye color. And the kid ends up with the dominant eye color. This doesn't always happen, but it's just one little example. Something important to remember is that dominant doesn't mean it's the most common trait. You don't have to write anything down from this slide. But one disease, Huntington's disease, we'll talk about that in a bit, the gene's dominant, meaning you only need one copy and you have the disease. But Huntington's is not very common. So just because it's dominant doesn't mean it's like the most common trait or gene out there. Now, a recessive gene or a recessive trait only shows up if there's no dominant gene. And we use a lowercase letter for this. Recessive, this would mean that there were two mutated genes in order to have a disease. So remember we talked about cystic fibrosis? Richard had two mutated copies. He had two mutated copies of the gene and therefore he had the disease. So sometimes diseases you need two copies of that gene because it's recessive. So let's do a little practice here. Brown eyes are dominant to blue eyes. The first thing we always do is assign a letter. Capital B is brown, lowercase b is blue. Okay. So if we have two big B's, remember one from one parent, one from the other parent. So every individual always has two. Okay. Brown eyes, big B would be brown. What about big B, little b? That would also be brown because capital letter is dominant. It's going to quote unquote win over. And little b would be blue. Okay. Now we have some other words we have to talk about as well, because you're going to see these words in word problems. Okay. So one of the words here, there's two different ones, homozygous and purebred, and they mean the same thing. I most often see homozygous. I don't often see purebred, but we have both of them in there. Okay. The prefix homo means same. Okay. So these are two of the same genes or same letters. So if I say in a word problem that somebody is homozygous, it means either they've got two dominant, two capital letters, or two recessive, two lowercase letters. So we can write that down. And if you don't want to write homozygous dominant, homozygous recessive, you could just write big A, or little a, little a. So if you see that it says homozygous, it means they either have to have two capital letters or two lowercase letters to symbolize that gene. On the other hand, if we have heterozygous, okay? Heterozygous is the opposite. It means two different letters. So heterozygous or hybrid, like a hybrid car, runs on gas and electric. Two different genes or two different letters. So big A, big B, little b. So in these word problems, when you're solving them, if you see the word, oh, the one parent is heterozygous, right? You know, oh, that means they have got two different letters. So let's do a little practice again, okay? In this example, it says green leaves are dominant over yellow leaves. So we got big G, green, little g, yellow. So I want you to think to yourself, what letters represent a homozygous dominant individual? Homozygous dominant. Remember, homozygous means two of the same. Dominant means the capital letters. So this would be big G, big G. Homozygous recessive individual. Homozygous means two of the same and recessive, so this would be little g, little g. Heterozygous means one of each, so that would be big G, little g. All right, two more words we have to do. Okay, and those two words are phenotype and genotype. At the end of these problems, they're going to ask you, hey, what are the genotypes or phenotypes of the kids? So you have to know these words in order to be able to answer those questions. So if at the end of the problem it says, what are the phenotypes of the kids? We're talking about the physical appearance, okay? So what they physically look like. Whatever their traits are. It could be a disease, could be their eye color, hair color. If we're talking about genotypes, we're asking, well, what are the possible gene letters that the kids could have? So it's two different things there. So let's do a really quick problem, okay? We call these monohybrid, mono meaning one, okay? Hybrid meaning two different parents, basically. So it just means problems with only one trait. You have the 7 steps, okay? And you always have access to these 7 steps when you are doing these genetics problems, okay? So you always have access to them. You don't have to memorize them. So first thing we have to do is identify the dominant and recessive genes, okay? So in this case, it says brown eyes are dominant to blue eyes. Cross a hybrid brown-eyed male with a heterozygous brown-eyed female. So it told us in the first sentence, brown eyes are dominant to blue eyes. So that means for dominant, I'm going to go ahead and write brown eyes. For recessive, I'm going to write blue eyes. Step 2, determine what letters will represent the dominant and recessive genes. So we need to assign a capital letter to the dominant gene and a lowercase letter to the recessive gene. So we're going to say brown eyes equals capital B, blue eyes equals lowercase b, or you can use any letter you want. So actually, I didn't end up using B. I used A. You could use Z if you want. The only thing I'll say is you want to make sure that your capital and lowercase look different. For example, if I use P, big P and little p look very similar. So that one might be a little bit confusing to use. Okay, so we've got A and A. Step 3, determine the parent's genes. So it says a hybrid brown-eyed male. Okay, so we've got hybrid brown-eyed. So hybrid means one of each letter. So we're going to have a big A and a little a. And heterozygous. Hey, that means the same thing, big A, little a. So we have our parents' genes. Also, if you're following along, there's nothing to write in the note packet or your notes. We are just doing this problem together. Sorry, I didn't say that. All right, then we've got to determine what genes go into each sperm and each egg. And this can get a little bit tricky, okay? So we have an activity in class to help demonstrate this, but I'm going to give you the short version of it right now. Okay, so basically, when we're creating sperm and eggs, Remember from meiosis, okay? In meiosis, we create eggs, sperm and eggs, with half the number of chromosomes. Okay. So I want you to take a look. Here's an activity we did in class, okay? If our parent cell is big A little a, and our other parent cell is big A little a, we need to figure out what combinations can go in their egg cells and their sperm cells. But remember, they only give half to the egg, and they only give half to the sperm. So what we end up getting, okay, is we can drop our letters down here, and our egg cell could either have a big A in it or it could have a little a. It can't have both because remember, half the number of chromosomes is our starting cell for meiosis. Same thing for our sperm cells, half the number of chromosomes, okay? So what we're actually doing when we have a Punnett square then, which we'll do in a minute, in these genetics problems, here's our goal, is to figure out what combinations, how many different combos of an egg cell and a sperm cell can I get? So notice I could put the big A and the big A together. That's an egg and a sperm. I could put the big A and the little a together. That's an egg and a sperm. I could have this little a and this big A. That could go together. And this one and this one. So it's 4 different combinations. And that is what our Punnett square is going to help us with. And if you're like, Ms. Franks, I never heard the word Punnett square before, don't worry. I'll tell you what it is in a second. But our whole goal is to figure out what combinations of eggs and sperm could we have, because we want to know the likelihood that a kid, an offspring, is going to have a certain trait. All right, so back to step 4. It says determine the genes going into each sperm and each egg. Here's what we do. We draw the little sperm, okay, and we put 1 letter in each. And then we draw the eggs, and again, we put 1 letter in each because half go into each of those cells. Step 5, add the sperm and egg letters around the Punnett square and solve the Punnett square. So here's what's going to happen. I don't care if you put the male or the sperm donor on the top or on the side. You just have to promise me you're going to keep the letters together, okay? So in this case, I have them on the side, 1 letter by each box. And then the female, 1 letter on each side on the boxes. Again, you could switch them, that's fine, as long as you keep the females on one side and the males together on the other side. So after we do this, we're going to drop down the letters, okay? So in this box, I'm going to have a big A and a big A. This box, I get a big A from this side and a little a here. Here I've got a little a and a big A. Here I've got a little a and a little a. So I'm going to fill them in. What this represents is exactly what we did up here where we were figuring out what combinations of sperm and egg could I have. Okay, we just put it in this box. So these represent the kids, the offspring. So my next question is, how many will have brown eyes? Remember, brown was dominant, symbolized by a capital letter. So how many of these squares have a capital letter? Well, 1, 2, 3. 3 are going to have brown eyes. How many are going to have blue eyes? 1. Now, this doesn't mean, oh, you're going to have 3 kids and 3 will have brown eyes and 1 will have blue eyes. These are the chances. And so we can write it as a percent. 75% chance of having brown eyes, and this should say 25, that's a typo. 25% will have blue eyes. Okay. We know this doesn't always happen because you know families where maybe they have 5 females, okay, 5 girl kids, okay? And we know that it should be 50% boys, 50% girls, but that doesn't always happen. The other question you might get asked is the genotype. That's our letters. So how many have the genotype big A, big A? Well, we just did to count the boxes. 1. 1 out of 4, or 25%. What about big A, little a? 2 out of 4, 50%. What about little a, little a? That is 25%. So that's how you do Punnett square problems, okay? Your sub is going to pause the video right now, is going to hand out your worksheet, And I'm going to walk through the first problem with you on your worksheet. All right. You're going to notice this is the same problem, but your worksheet looks different than this. Your worksheet looks really blank. That's okay. I'm going to do the online version because I don't want to have to try to hand draw in a Punnett square right now. But your worksheet, it's the same thing. It's got the same problem here. Okay. The only thing is I just have some extra words. Okay. So the first thing we need to do, step number 1, is figure out what's dominant and what is recessive. So it says in a certain plant, long stems are dominant to short stems. So that told us right there. If it didn't tell us, one thing we can do is if it says heterozygous long-stemmed, we know heterozygous means one of each letter. And so the dominant trait's going to win, all right? So if it's heterozygous and it says it's long stem, that means long stem must be dominant. That's another way you can look, okay, or check. So long stems are dominant to short stems. So the dominant trait's going to be long stem. So on your worksheet, I would like you to write on next to number 1, dominant trait equals long stem. You can just write dominant equals long stem or long. Recessive equals short stem. Now we have to assign letters. We need the letter representing the long stem. So we want to pick a letter. If you want to choose big L, that's fine, as long as you make your little l look different. Okay. A lot of times people pick the first letter of the dominant word, and that totally works. Again, just beware of stuff that looks the same. Like for example, if you use big O and little o, they look really similar. Big L and little l look enough different that I think we're okay using them. So on your worksheet, number 2, you can write, again, if you want to abbreviate this, you can just write long stem equals big L, short stem equals little l. Now we got to figure out the parents. Okay, so it says that one parent is short stemmed. Well, we know short stem is recessive and it's little l. Well, if it's recessive, it can only have 2 little l's. So we're going to write little l, little l. So you can write parent 1 equals little l. You can write mom equals little l, dad equals little l, whatever one you would like. Or you can just write genotypes of the parents equals and write both of them, okay? And then the heterozygous long stem. So heterozygous, that means one of each letter. So that would be big L, little l. Now, on our PowerPoint, on our Google Slides, we saw that we draw the actual sperm and the egg. So instead of my lines here, here's what I would like you to draw. Can you draw 2 circles underneath the little l, and 2 circles underneath the big L, little l? And what I want you to do is write one of each of the letters in there. These are going to represent the sperm and the egg. So you can actually, if you want, you can go, let me go back to our slideshow here. This is what it should look like. I'll move my screen over a teeny bit here. So you can draw 2, we're not going to use these letters, but 2 sperm and then 2 eggs. Okay? And inside each one is going to have 1 letter. So I'm going to have a little l and a little l and a big L and a little l. Again, you're doing this. You have circles around yours. I don't because I'm on a computer. Now, the sperm go on one side, the egg go on the other side. Okay, so I'm going to put a little l here, a little l, and I'm going to put a big L up here and a big L up here. And then I'm going to fill it in. So here I'm going to have a big L and a little l. Now, I will tell you, typically we always write the dominant letter first, okay? It's not wrong if you write little l, big L. It's just we usually always write the dominant letter first. Here I've got a big L, little l. Oops, sorry. That would be a mistake. I have a little, 2 little l's. Big L, 2 little l's. There's my Punnett square. So then we need to figure out what's the chance of having a long stem. Okay, so I've got 2 that are big L, little l. And I know that big L is long. So that would be 2 out of 4. And 2 out of 4 is the same as half, so that would be 50%. What's the chance of having a short stem? That's little l. That's 2 out of 4, or 50%. Next one, how many genotypes exist? Genotypes are different gene letter combinations. I have a big L and a little l, and I have little l. So I just have 2 different letter combinations. So 2. And we're going to list them. I got big L, little l, little l. How many phenotypes exist in the offspring? That means how many physical traits, how many different appearances will I have? Well, I know some are going to be long, and some are going to be short. So that's just 2. And I have long and short.